That my wife wasn't here to hear all those wonderful remarks you made about me.
Oh.
I never hear that from you.
Well, we've got them on tape.
Yeah, you've got them on tape. Please send them to her. She needs to enjoy those. So I'm going to try very quickly and go through with you the combination therapies enhancing the prospects of curing diabetes. The background to this was that saran wrapping reverses tryptophan-induced diabetes in hamsters. Note, this wasn't the 300 mice you heard about. These were hamsters, actually. So I don't know if they come into the same category. We got a crude extract out of the rat's pancreas, and that we called inotropin to move the islets. And this crude pancreatic extract from the cellophane rat, that's cellophane, but we called the discovery serendipity because it fits more with the accidental discovery. And this induced new islet formation from ductal epithelium. Inotropin itself, can I go back up? Where's the up button? Yes, inotropin itself reversed tryptophan-induced diabetes in hamsters, and mRNA differential display led to the discovery of the major gene that was turned on over here, out of 28 genes that we uncovered, which we called INGAP, islet neogenesis associated protein. The people involved in this over here, I'll just show you very quickly. This is Renette Rafaelov, who's now Renette Fail. I thought she might be here today. Scott Barlow, Gary Pittinger, and I'm sorry I didn't have a picture of Laurie Rosenberg. He was involved in the discovery as well. Now, what transpired from this is we were able to clone and sequence the gene, as Rita has shown you so far, and it's a fairly large gene with 766 base pairs, but we were much more interested in trying to get a biological core out of this protein that could do what the whole protein had done in the case of inotropin, or in the case of the protein expressed from the gene itself. So we found in the mature peptide a region from amino acids 104 through 118, 50 amino acids that was capable of duplicating everything we'd seen with inotropin, thinking that this would now then be able to cure diabetes. To show you very quickly, a study published by Lawrence Rosenberg, myself and colleagues back in 2004, is if you took C57 black diabetic mice and you didn't treat them or treated them with saline, their blood glucoses ran around 400 milligrams per deciliter. But if you gave them INGAP, you restored the concentration of blood glucose to normal, and if you stopped the INGAP, it persisted, saying that a biological effect had been achieved. This wasn't simply an effect of INGAP itself stimulating insulin secretion and reversing the diabetes. Biology had changed. Yeah. The question is, what was the biology? Here you see a control islet. That is streptozotocin administration. You see disappearance of that islet with an inflammatory cell infiltrate into the pancreas. Here is if you add the INGAP to that, you see here a pancreatic duct, and here growing off the duct, this whole new islet with expression at that time of what we'd shown of 4 different hormones that were produced. So once you initiated the process, it made the hormones that you wanted to have, including glucagon, which was counter-regulatory, which we also wanted to be produced. You see too that with the INGAP over here, you see a huge reduction in the inflammatory cell infiltrate, and that brings up another issue as to whether or not it has the capabilities of reducing inflammation and immune tolerance. Everybody said, of course, this was hamsters and it was into humans, so we now had to say, well, was this indeed present in humans? So is INGAP present in the human pancreas? Does INGAP affect human pancreatic tissues in vivo and in vitro? And will it reverse diabetes? Well, the questions now. And this is a most interesting example of what can happen. If you take islets from humans and you have them in tissue culture and you add INGAP to them, nothing happens except you stimulate insulin secretion. However, if you allow those islets to continue in the incubation, the human islets after isolation become CK19 negative, which means that CK19 is a ductal antigen, and now you have transformation to CK19 positive ducts over here in culture. But now if I add INGAP to those ducts, not the islets, now what happens over here, there's transdifferentiation of these ducts to begin to form what you see here shown as a new islet within the wall of the duct expressing insulin. If we take that a little further, the INGAP peptide induced transition from ductal cells to insulin and fully formed islet with expression of insulin, which I'm showing you here, also expresses glucagon, somatostatin, pancreatic polypeptide. But I hadn't at that stage looked for ghrelin expression.